record on this computer. Awesome. Thank you everyone for coming. Oh, one more person has entered. So today is our third day of STEMTober, our third weekend. For everyone who hasn't been with us so far, STEMTober is hosted by Wave Learning Festival and STEMI. Wave Learning Festival is a nonprofit educational platform that offers free courses for middle and high school students by college students in an effort to improve educational equability. And STEMI is a nonprofit organization that also hosts STEM classes and events primarily for middle and high school students. So every Saturday in October, we'll be focusing on a different STEM field, relatively different for interested students who maybe have not had as much exposure to STEM in their schools. And weekend one is all about engineering. It's up on our YouTube channel. So if you didn't get to watch any of that, you can go see it on our YouTube. Weekend two was on computer science and math. That will also be on the YouTube channel soon. And weekend three, which is this weekend, is on medical science and healthcare. Weekend four is going to be our last weekend of STEMTober. That's on natural sciences. So specifically, we're going to go into chemistry and physics primarily. So now I'm going to introduce our first lovely speaker. Thank you so much for joining us. Dr. Shaun Brinsmade is an associate professor with the Department of Biology with a joint appointment in the Department of Microbiology and Immunology at Georgetown University. And today we'll be discussing the role of bacterial behavior during infection. So please, everybody, welcome Dr. Shaun Brinsmade. And Dr. Brinsmade, you can take it away.
Thank you so much, Diana. I just want to take the opportunity to really thank Diana and the other organizers for the Wave Learning Festival and for STEMI for the invitation and for giving me the opportunity to talk with you all today. You know, one of the great things about my job is that I get to go and travel and talk about the work that our lab does. You know, I get invitations from my colleagues and other faculty members across the country, but it really is a special time and a really great opportunity when students invite me to give a talk. It's got special meaning to me. So thank you so much for the opportunity to talk today. So I'm going to try to share my screen now. Hopefully the tech works. And Diana, if you don't mind, when I share my screen, I can't see the chat or anything. So if people should feel free, if they can, to type in any questions into the chat, and I'll be happy to try to answer them. I'll try to have a couple of pauses in between when I talk today. So I'll try to address them. Otherwise, you know, I think I'm going to leave enough time at the end so that we have a chance to talk to one another a little bit more. All right, so let me just switch my screen. Let's see how this goes. Oh, Diana, the host apparently has disabled attendee screen sharing. Can you enable it for me?
Sorry, I was on mute. It should be enabled now.
Let's see.
Awesome.
Can you see that?
Yeah.
Perfect. Awesome. The tech works. We'll see if it continues to stay that way. All right. So let's go. So today I want to tell you a little bit about some of the work that we've done and kind of where I've come from and where we're going in the future. As you can see, at least later on in the talk, we're going to talk a little bit about a bacterium that's near and dear to my heart, not because because I've been infected, but because I actually use the bacterium to understand biology a little bit better. And that's the bacterium and the pathogen Staphylococcus aureus. So let's see, let me go ahead and switch. So this is just a kind of an outline sense as to where we're going to go today. So I'll first talk a little bit about me and where I've come from and how I ended up at Georgetown. And then I'll talk a little bit about the science that we're doing in the lab at Georgetown in Washington, D.C. And then I'll offer some parting thoughts about careers in science. And then I hope to hear from you all too. So a little bit about where I've come from. So I was born and raised in Shelton, Connecticut. So that's in New England, in a very small town. There's really no claim to fame other than the wiffle ball is manufactured there. So if you've ever played wiffle ball outside, I don't know if you all today do that. At least it used to be popular when I was a kid, but I'm a little bit older you now. But Shelton is about an hour and a half from New York City, so it's easy to get to the city from my town. I tried to get into the city when I could when I was younger. And I went out through high school, Shelton High School. I was a band nerd. I don't know if anybody is a band nerd there. Oh, I see Diana was waving her hand, so awesome. I'm a trumpet player, but I always loved my time there. I met my best friends there. And I went on after Shelton High School to the University of Connecticut, where I was also in the Yukon marching band there. But I was a molecular and cell biology major there. And so I always have to have a shout out to Go Huskies. That's our mascot. And so shown over here, I don't know if you can see it, but this is a kind of a nice, beautiful shot of campus with the dome where the Yukon women's basketball team used to play. I'm not sure if they still do that. But I really enjoyed my time there at Yukon. And I didn't actually expect to be there originally. I had my heart set for a number of reasons on a different university, which I won't name. But at the time, my family didn't have a lot of money, and we didn't, I couldn't afford the school that I originally wanted to go to. But the University of Connecticut was my eventual choice. And actually, I have to say that I had so many experiences there that actually were really pivotal in my development, if you will, as a scientist. I had the opportunity to work with a fantastic biochemist when I was there. I actually learned about it when I was in my biochemistry lecture, and there was a call for undergraduate students who were interested in doing scientific research to talk to a particular faculty member, and that happened to be my future undergraduate mentor, Dr. Judy Kelly, who I've circled there. She's since retired now, but I owe a lot of my my early years to science to her. She studied the structure of different proteins and trying to understand how the structure of a particular protein conferred certain activities and a function to those proteins. And to do that, she used a technique that I don't really want to get into too much, but it uses X-rays and a little bit more powerful form of X-rays to determine protein structure. And the only other thing I'll say, right, is that she believed in me as an undergraduate, and a junior scientist, so much that she took me to what is now Brookhaven National Labs on Long Island in New York. And that's a national laboratory that actually uses high energy electron beams that are actually generated in this circular building to generate the energy that's required to probe protein crystals that allow for structure formation. And I'm just showing you here that my hard work actually resulted in a paper when I was an undergraduate, and that kind of helped spur my continued interest in science. This is just a little bit more, and kind of just as a shout out to let you know that very early on you can get involved in science and very important scientific research. And so, as I mentioned, I worked with Judy Kelly to do crystallography, and that essentially, if you've ever had rock candy, you essentially do the same thing. You take proteins and you slowly crystallize them out of solution. And so they make these beautiful crystals that you can see here. I think you can see them. They're not actually green, but that's just the way that the color, the filter on the microscope was being used at the time. But you shoot X-rays at the crystals and you get these interesting patterns. And you can hear my dog in the background now. Hopefully he'll stop barking. But anyway, I just wanted to show you that this is me right here when I was not too much older than you are now. And Judy Kelly and her research team allowed me to help them to understand protein structure and function. And this is just a figure from that paper. But I don't want to dwell too much on that because that's kind of old news, right? But I wanted to also mention that when I was at UConn, I had a number of other faculty members, professors, teachers, and mentors that were really important in driving and kind of steering me in the direction that I eventually went to. Excuse me, I have to move backward 1 second in time here. Yeah. And one of those is, actually he's since passed away now, but he was a dear friend of mine, Dr. Ed Ledbetter at UConn in the Department of Molecular and Cell Biology. And he taught a microbial diversity and ecology class. And he taught in the style that he was brought up on. And so shown here on the right-hand side here is a gentleman by the name of Dr. Van Niel. And he was one of the first microbial ecologists. So you might be familiar with ecologists that go out and they can survey the different types of birds or animals or plants that are out in nature. But Dr. Cornelis Van Niel was interested in all the different types of bacteria and other microbes, fungi, and the other single-celled organisms that were all over the place on the planet. And he was kind of one of the first ones to recognize the field of microbial ecology, and in fact founded it. And he had the belief or hypotheses that microbes are found everywhere, and that every molecule in nature can be used as a source of carbon and energy by a microbe somewhere. And in fact, I carry this on to this day, and I will tell you that I've learned through my own experiences that microbes are the best chemists. So that might be kind of a weird thing to think about. I tend to anthropomorphize bacteria because I was kind of brought up to think about how cool bacteria were. So don't be afraid to do that if you feel really passionate about something. But I think they are the best chemists. They can do really cool chemical reactions that we can't even think about doing ourselves. And sometimes we need very harsh conditions to do them. Anyway, these microbes are fantastic and they can do really cool things and live everywhere. And Dr. Ledbetter was really instrumental in shaping my understanding of microbes, because he used the same technique as Dr. Van Niel in the classroom. And he used to, so both of them would talk about all the amazing capabilities a bacterium could do in terms of growing and living and doing chemical reactions. And we would talk in class about how potentially the bacterium might do this and how you might be able to take advantage of that information to get your hands on it in the lab, right? To play with it, to purify and isolate the bacterium. And so they, Dr. Ledbetter would kind of, steer the conversation a little bit, say, where your logic might be good, where it might not be good, giving you tips as to, how do you isolate these bacteria. And then at the end of that discussion, right, we would go into the lab and then we'd actually try to isolate the bacteria from different samples. And I can tell you, I've isolated different types of microbes and bacteria from cheese, from the soil, and even from my own mouth. And I can tell you that some of the stinkiest bacteria that I've ever found were isolated under special conditions from samples from my saliva. So it's really cool. It opened my eyes to the world of microbiology. And in fact, one of my graduate teaching assistants that helped Dr. Ledbetter in the lab, the teaching lab when I was a student, really told me that if I wanted to pursue microbiology further, I just had to go to the University of Wisconsin-Madison. And that was one of the biggest places or best places to go for microbiology. And so at that time, I was a senior in high school, in college, and I wanted to learn more about it. So I went to the University of Wisconsin for my graduate work. And so then I changed my allegiance. So now instead of being a Husky fan, I was now a Badger fan. Still am a Badger fan. When I went there, I joined the lab of Dr. Escalante, Jorge Escalante. And I should note that when I got there, I actually thought that I wanted to continue doing the X-ray crystallography, the protein structure stuff that I did when I was an undergraduate. I thought that was the coolest thing. And Dr. Escalante actually was not a crystallographer, but he tried very hard. And I test drove his lab. I went in and I did some experiments, as a lot of young graduate students do. And I kind of got bitten by the bug, so to speak, in that his lab studied the inner workings of bacteria and enzymes and kind of the biochemistry and genetics of bacteria. And that was a complete 180 from what I did as an undergraduate. And this is kind of a lesson to you all. And that is keep an open mind whenever you're trying something new, because you never know what's out there unless you try it. I did not know that the type of research that Dr. Escalante was doing, I didn't know it existed. And because I thought, I was going to do crystallography, but once I tried it, I never looked back. And I have to, thank Dr. Escalante for a lot of the work that he did. And I guess the bottom of the screen here, this is just kind of one of the perks of being at the University of Wisconsin-Madison. People think of Wisconsin as being cold, inhospitable. In fact, it's a lovely place. It's hot and humid in the summer, but you can't beat the sunsets at night. And the university is right on a lake. So I kind of had some of my best times with my fellow graduate students on that terrace, eating dinner and, listening to music at night. But when I was a graduate student, I focused on an enzyme, so of all the things that you could study, right, I picked one enzyme in one particular bacterium, and it turned out to be the food poisoning bacterium Salmonella enterica. And so the reason, I guess the reason, or maybe the question you're probably asking me is, well, why did I decide to do that? And I guess I've always been interested in how things work in a cell, right? So even from when I was a little kid, I'd pick up a leaf and I'd see, look under a very crude microscope in class, what looked like cells in a leaf. And I was always fascinated in how, in a tiny little space like the cell, how could light be shining onto the leaf, and how does that light get converted into energy, and how does that help the leaf and the plant that it is to grow, right? And how does it make all the food, right? Fixes carbon. So I've always been interested in kind of how things work inside a cell. And one of the important things are that in a cell are proteins and enzymes. And so I studied this one particular enzyme, and I'm not going to get into all of the nitty-gritty details of that. But after about six and a half years worth of work in working on this enzyme, and the enzyme is called PTA, or phosphotransacetylase. And you don't have to take much away from this other than the fact that this is an enzyme that's really important to bacteria, in that it takes a sugar or a carbon source, a food source, and converts it it allows it to convert it to a compound that allows it to generate energy. And that in itself is kind of interesting. But the cooler part, and this is where I came in, is I discovered that enzyme was really more clever than we thought, in that it would actually pay attention to the different levels or concentrations of different chemicals or metabolites in the cell, right? So we talked about sugars and other molecules that are in the cell. Well, some of them actually tell the enzyme to either speed up the reaction or slow down the reaction. And that allows the cell then to decide whether the substrate for the reaction, right? So that in this case, it's acetyl-CoA, that acetyl-CoA can either go into further manipulations in metabolism or can be converted to a different compound by my enzyme. And so those particular compounds, right, are ATP and NADH. And I don't know whether you've learned about them in much detail in your classes, but these are really molecules that are important energy carriers in the cell. And so at the end of the day, what we learned and what I discovered is that this enzyme was helping the bacterial cell to optimize its metabolism based on the environments that it found itself and the conditions it found itself in. And it was using these metabolites to, whoops, sorry, back up 1 second, if I can. Doesn't want to back up. Okay. And what it did is it allowed the bacterium to make a decision as to how to redirect molecules in metabolism. And I thought that was really cool, right? That there was actually information provided by these small molecules in the cell. And so around that time, right, I was finishing up my graduate work, and a person who turned out to be my future mentor and postdoctoral advisor, Dr. Link Sonnenschein, came to give a talk on another protein that he was really excited about when he, right around that time, and his lab had discovered this protein called COD-Y. And this is a protein that is not an enzyme, so it doesn't convert substrate to product in metabolism. This is a protein that actually can turn genes on and off to alter the levels of proteins and enzymes in a cell. And again, this is a bacterial enzyme. And one of the things that Dr. Sonnenschein was really excited about when he gave his talk back when I was a senior graduate student was that this is a protein that he discovered in a soil bacterium that you can find outside your apartment or your house or wherever. This is a protein that allowed the bacterium to turn on and off the various ways for the bacterium to obtain food or to swim away to find new nutrients. Okay, and that in itself is kind of interesting, but there was a common theme that I picked up on when I was listening to his talk, and that this protein turned genes on and off by sensing certain molecules in the cell. So in this case, certain amino acids that are the building blocks for proteins, and certain energy molecules like this one, GTP, which is important for protein synthesis. And so if you, and I guess at the end of the day, with the way that it works is when this protein senses that amino acid or GTP, the shape of that protein changes and it allows it to bind DNA. And when it does so, that allows the protein to either turn on or turn off a particular gene of interest. Okay. And so if you're following along, you might detect that there's a common theme here. And that is that this is another example of how the bacterium uses information provided by small molecule chemicals or metabolites in the cell to make decisions as to what to do. And so after hearing about this really excited me some more, and Link, Dr. Sonnenschein, to convince me to come to his lab to work on this protein, COD-Y. There was still a lot to learn from it. And in particular, what we learned is that not only is it found in this bacterium that you find out in the soil that is, doesn't cause harm to anybody, but it's actually also found in some bacteria that cause disease that you might have heard of, right? So you've heard of MRSA, which is a particular form of Staphylococcus aureus. That this is a drug-resistant form of staph. You get it when you get cut and you get a skin infection, oftentimes. But it's also COD-Y is also found in bacteria that you might hear about in the news. That's sometimes weaponized, anthrax, for example. And then also organisms that cause intestinal diseases, as well as respiratory infections as well. And so this meant that codY actually was playing a bigger role than we thought. And I'll get to that more in just a second. So I'm going to take a pause now and let you know that essentially after all of this, so my days at UConn, my days at University of Wisconsin, and my days at Tufts, where I was working with Dr. Sonnenschein, all trained me to be what's called a bacterial physiologist. And I'm curious now as to know if you know what that is and what that might entail. So I'm going to try a little experiment right now, and I don't know if it's going to work, and if it doesn't work, that's okay. And so I think, and it's a little bit different from my screen, but I think it might be at the bottom of your screen. There might be an option to look at, there might be a button to push called annotation or annotate. And at least for me, if I push that button, there's a menu will pop up on the screen that has what's called a stamp. And I'm just going to show you right now that if I select a stamp, and I have different options, for example, a star here. And if I now click somewhere on my screen, I can put a stamp down, and I see somebody else has put something in. Good, so there's a check mark there. And you can change it. There's an X and there's a heart and other things. So what I'm going to do is I'm going to clear this. I'm going to clear everybody's drawings. So stop drawing for a sec. And now I want you to vote by putting your sticker or your stamp next to these three options. So do you know what a microbial physiologist is? And so you can stick it on either yes, no, or maybe based on what I've already told you. So go ahead and take a moment and vote as to if you know what a microbial physiologist is.
For anyone who's having trouble with the annotate, if you see, it should say at the very top, you are viewing Shaun Brinsmade's screen, and then it says view options. If you look at that drop down, it should give you annotate. Let me know if that doesn't work. Oh, okay. I see it is working.
It looks like some people are voting, yes.
Oh.
So thank you. So I'll just give it a couple more seconds for those to respond. And so maybe you do. This is kind of fun. I like using, one of the best things about the whole situation now of the pandemic, right, is I've had to learn how to teach virtually in my classes at Georgetown. And so I've kind of become more familiar with how to interact with people from a distance. So thanks for your responses there. You'll have another opportunity to play with this a little bit later. So I'm going to clear these. And so generally, this is exactly what I suspected. And perhaps you don't know what a microbial physiologist is, and that's okay. That's why I'm here. So I'm going to clear those. I'm going to stop. And so what a microbial physiologist or a bacterial physiologist is somebody who's interested in the inner workings of bacteria. How do they tick, right? So what if you were to basically if you're a car fanatic, for example, you would look under the hood and you would tinker with things in the hood to figure out how your engine works, right? And so that's kind of what I do as a bacterial physiologist. I essentially use various biochemical and genetic tools to understand how a bacterium works. So I'm essentially peeking underneath the hood of Staph aureus and other bacteria in the laboratory. And essentially, this is how bacteria do various things, how they eat a various food source, how they multiply, and how they grow. Because every bacterium wants to become bacteria, right? More than one bacterium. And what's further is that there are other processes that we can study. For example, my lab has studied in the past how bacteria actually work. So bacteria are single-celled organisms, right? And so you can see some of them down here. So these are electron micrographs of Staph aureus bacteria, and each circle here is a bacterial cell, and they act as single units. But every now and again, they actually can coordinate their activities They can talk to each other using a chemical language. So they can sense one another, and they can do things that they would rather do as a group rather than individual bacteria. And so one of the things that they do is when they can come together, and then they can create a sticky little glue that allows them to come together and almost act like a multicellular organism like you and me. And that's what's shown here on the left-hand side, right? This is how they actually develop what's called a biofilm. And there are multiple stages of that, right? And these are three-dimensional, almost like mushroom structures, right? And this is where bacteria actually can survive very harsh environments. And in fact, in biofilms, bacteria are more resistant to antibiotics, which is a problem. And so that's one of the reasons why we like to study biofilms, is if we understand how they work and how they're formed, we can figure out how to get rid of the biofilms and help patients in hospitals who have infections that are biofilm related. Okay, so that's bacterial physiology in a nutshell, and I'm happy to answer more questions later on it. But so I want to fast forward a little bit more to now. So I came to Georgetown University in 2013 in August of that year, and so shown here, this is kind of the trademark building, if you will, right? So everybody's postcards that come from Georgetown have this image on it. This is Healy Hall, the famous clock tower. And actually the president of the university works in that building. My lab is actually in a building called Regents Hall. And actually my lab is on the other side of that building, but my office is kind of right here. So if you email me during the day, you'll be sending your emails to my computer in that room. But at Georgetown, I've been there for now about, I guess this is about 7 years now. And I established a laboratory where I essentially have a team of researchers working with me to solve really interesting biological questions. And actually, this is a little bit older picture now. Some of these folks have left to go on to other things. And I'll get into that a little bit later. So I have a couple of, so actually this is Alyssa, who is a graduate student in my lab, Kevin Malinik, who is a graduate student in my lab. Both of them have since graduated and now have moved on to have other research careers. And I have a current PhD student in my lab, Shahad Al-Khatani. And then you'll see some other folks in the lab. This is Sarah, John, this is Stephen, Carson, and others, and they are undergraduates that are working in my lab. So these are not that far. So remember when I was a crystallographer, these are folks that are actually of that same age, and in fact, not that far off from where you are right now. And we're all working together to understand how Staph aureus and other bacteria work. They're all, I would consider all of us bacterial physiologists. And the question, one of the main questions that drives our research in our lab is, how does a bacterium's environment control the bacterium's behavior? And again, behavior is kind of broadly described, right? We can talk about how it forms a biofilm, for example. But what about the ability to cause disease, right? You can imagine that it might be as a behavior. And so we're interested in how, let's say, the host environment, how if a bacterium gets inside of you or on you, right? How does that affect the bacterium's ability to cause disease? And we work with Staphylococcus aureus, which you may have heard of as a nasty pathogen. It turns out that this bacterium is can live on you as a normal component of your what we call the natural flora. And but at some point along the way, it turns from being kind of a silent hitchhiker on your body to one that causes disease. And in fact, it is actually the #1 cause of skin infections. It's resistant to multiple antibiotics, so that's a problem, right? Because if you get infected, you need to treat the infection, and sometimes your infection isn't solved and resolved by antibiotics, or at least the ones we go to first. And while there's been a lot of work, really good work that's been done to reduce how Staph aureus causes infections in hospitals, when bacteria would actually kick you while you're already down, while you're already sick, we're actually seeing that there's actually a rise in what we call community-acquired Staph aureus infections that are multi-drug resistant. And so, in fact, Georgetown had a slight problem with that a few years ago with their in their dorms. And in fact, this is a problem if you're an athlete. If you're an athlete in high school or in college, you're coming into close contact with other individuals, and you can transmit this bacterium from one to another, from hand-to-hand touch, for example, or getting it from a surface like, I don't know, your exercise mat or your weightlifting dumbbells, for example. And so it's a real problem, and we need to figure out how to solve this problem, because it costs a lot of money to the healthcare industry, and it causes a lot of, it's costly in terms of your human health too. And so we're trying to come up with new strategies to understand how the bacteria work and how to eradicate these problems, these pathogens. And I just wanted to mention that this is a pathogen. In this case, is one that we talk about, an opportunistic pathogen, but there are pathogens that have no business being on you or in you, and those are what we call primary pathogens. But we're really talking about these secondary pathogens like Staph aureus. And I'm going to talk about what's called pathogenesis. And this is just to keep in the back of your head, this is just the ability, or pathogenicity is the ability to cause disease. Okay, so when I say pathogenesis, I'm going to say the development of the ability to cause disease. So think of it as kind of a computer program, right, in the bacterium that turns on, that allows it to cause disease. And it's actually composed of both how infectious a bacterium is and how virulent it is. Okay, and these are based on the sort of tools that the bacterium has at its disposal to cause disease. And Staph aureus, I'm going to tell you right now, is a really clever pathogen. It can colonize essentially every surface in and outside of your body. And it does so by producing lots of different factors. And some of these are on the bacterial surface to stick to a surface, and others are enzymes that it can spit out. And those can interfere with your immune response, or can help slice and dice your tissue. And I'll get into that a little bit later. And I want to make sure I have a little bit of time here. But the point is that COD-Y, this protein I told you about before, is really important for turning on and off the expression of these genes, right, that basically allow the bacterium to cause disease. And I'm gonna skip over this in the interest of time. But so I want to get a sense as to what do you think? So use your annotation tools now, and I want to ask you, are bacteria our enemies? Do you think? So do you think that they mainly make us sick, or do you think generally speaking they get a bad rap, right? Or do you really not know? And that's okay if you don't know. So we got a couple of people who have said that no, they get a bad rap. And I'm going to call that done. So good, not all of them, thanks. And so I'm really, really proud of you all, because when I was young, I probably wasn't thinking about how there might be some bacteria that are actually useful to us. And in fact, the vast majority of microbes on this planet are beneficial or are not harmful, right? And it's only a small number of them, relatively speaking, that are causing disease. So thanks for your participation there. I'm going to clear these and we're going to move on. So you're absolutely right. Go ahead.
Oh, I just wanted to say some people also put their answers in the chat.
Oh, perfect.
Yeah.
And so are they all Are they yes or no, or I don't know?
Rasa said not always. Someone else said it depends. Some are harmful, some are not. And then same with our third answer there. Some of them are
Perfect.
harmful, but we wouldn't be able to digest food without bacteria.
Exactly. So I think the biggest thing that has come out of recent work, right, is this idea of the microbiome, and understanding that that you actually are not just a human with human cells, but you're a human because you have microbial or bacterial cells associated that actually do some work that, you know, for you that your body can't do, right? You can digest things, as somebody had noted. You can't digest certain compounds or foods, but your bacteria can, and then they break them down into smaller molecules that your body can actually use. Okay, so great. So thinking of that, right? I'm going to suggest then that, yeah, they get a bad rap, right? And so I want to ask you another question, and you don't have to respond to this, but I want you to think for a sec. What happens when you're hungry? How do you feel? And you might be thinking, well, actually, I get hangry, right? So I get angry and I'm hungry, so I'm hangry. So I guess what I would do is I would propose that actually bacteria also get hangry, right? And they get stressed out when they're in they're in an environment where they don't belong or where they don't expect to be, right? And they're starving for nutrients. And so in fact, all they want to do is to survive. And that I would wager then that this protein COD-Y that we study is actually really important as the hangry switch, and allows the bacterium to detect when it's hungry, right? So remember, when it's sensing these amino acids in GTP, so when it turns out that when COD-Y doesn't have those nutrients around to sense, right, it turns on the expression of these toxins and other factors that can help the bacterium to cause disease. And so and so I don't want to get into a lot of the data here, right? But what we found, right, is if you continuously starve the bacterium, right, or you make the starvation more and more severe, right, so they get hungrier and hungrier and hungrier, in a way, in a phenomenon that requires COD-Y. COD-Y is important for this response. Staph aureus, right, and other bacteria that cause disease, they sense that increasing hunger, and as they get hungrier and hungrier, they turn on more and more genes that are important for survival and that cause disease. And in fact, I'm going to draw your attention to this, right? So at the very end, when they're super starved and they can't possibly find more food, right, floating around them, that's when they turn on the production of toxins that pop open your red blood cells or your immune cells, or they turn on, you know, other enzymes, hyaluronidase or proteases or lipases. And what these do is they actually are enzymes that break down fats, proteins, and other molecules in your tissue, right? So this is the slicing and dicing that I told you about before. And so really what the bacterium is doing is creating nutrients, or trying to extract nutrients from your tissue to eat them. So this is really just the normal response of the bacterium to starvation. And so really they're not out to get you in many cases, they're just trying to survive. Okay. And so that's one of the biggest take-home points that I want you to understand is that the environment of the bacterium can control its behavior, and then that behavior is really just more of a kind of a self-defense mechanism, and that we kind of just get in the way. All right. And so then I guess the question then is, and I'll get to that in a sec, is, okay, you've got this protein called COD-Y that's sensing these nutrients, right? And that's fine, because so COD-Y can sense the starvation and turn on these genes, right? So how does that work? Does COD-Y do it itself? And it turns out that's not really the case in most cases. It turns out that COD-Y actually takes that signal, right, that starvation signal, and then passes that signal on to something else. Hence the trapeze work there. And that actually is me on the bottom here. That's another example of how scientists have a life, right? We're not robots that go into our closets at the end of the day. We actually have hobbies. So one of those is I do trapeze, which is kind of a crazy thing to think about, but yeah, I do it. It's kind of scary, I have to say, because I'm afraid of heights, but I kind of persevere. Anyway, so I'm running out of time, right? But I do want to tell you that we have found that there's another protein that allows cod-Y to work, or it helps cod-Y to help the bacterium decide how to turn on, if it's to turn on these toxins and other proteins to cause disease. Again, just to replenish those nutrients and foods that they're missing. And that is through this this protein system called the SAY2 component system. And unfortunately, I'm going to run out of time here. But what I wanted to tell you is that this is cod-Y controls the production of these proteins, and you don't need to know the names of these proteins, right? But they are important for turning on the expression of these toxins. And I'm going to show you just one experiment because I want to get to your questions that you might have. And so I just want to give you an example. So it turns out we wanted to know whether or not, whether or not cod-Y actually plays a role in toxicity, right? Conferring toxicity to your own cells, for example. If you get a staph infection, is cod-Y playing an important role? And so what we did, and nutrient starvation as well. So what we did was we did a simple experiment where we grew bacterial cells in a flask, and what we did was then we took, because a lot of these toxins are spit out of the bacterial cell, we got rid of the bacterial cells after they're growing, and then we used the growth medium, which presumably had potential toxins in it, and incubated that with neutrophils. And these are your neutrophils that you get from your own immune system in your blood. And we wanted to know what happened to these neutrophils. And so what we did then is in collaboration with a scientist in his lab at NYU, what we did was we did a cell death assay. In other words, we asked how toxic was the culture fluids from these bacterial cultures, right? Depending on whether cod-Y was there or not. So I'm showing you here and I just want you to focus on maybe this early part of the curve here. And so those open circles right there, these are the naturally occurring bacteria. And the other is that we have these black circles, which are bacteria that don't have cod-Y. We've engineered them to be missing that gene, and so therefore they don't make the cod-Y protein. And so what this does is this stimulates, this is similar, analogous to to a scenario where the bacteria are completely starved for nutrients. Okay, so think of this as completely satiated, and these guys are completely starving. And so I've made a big mess here, but I hope what you can appreciate is that there's a difference. There's a higher cell death, and this is neutrophil death, when cod-Y is missing. And so what that means is that cod-Y does control and suppresses the toxin production, right, when nutrients are abundant. But then when cells are starving, right, because cod-Y can't sense those nutrients, they turn on the production of toxins, right? And that's really a key finding here. And unfortunately, I'm kind of I want to save a little bit of time for your comments, so I'm going to save you a lot of the extra data that we have, other than the fact that I'm going to move to this, fast forward to here. We actually, I have to escape from here.
Dr. Brinsmade, I just want to say we have a break. We reserved a break from 1:30 to 1:45 before our next speaker. So you actually, if you're willing to, you can also stay during that time if you want to go over a little bit.
Okay. I wanted to be mindful of people's time, but
Yeah.
maybe what we'll do is I will kind of finish up here, and maybe if people have a couple other questions, I might be able to insert those data in there, but we'll see. We'll play it by ear. Let's see. I think what I'll do is maybe I'll use a couple more seconds of time. So, all right. So what I've told you thus far is that cod-Y is this important protein for controlling the production of these factors in a nutrient-dependent manner, right? The hangry switch, if you will. But then we also have a question in the lab, and so this is some of that work that was published. But we had another question in the lab, and that is, are all body sites created equal? So if you think about it, I said earlier that Staph aureus will colonize, and is very clever, will colonize essentially every site of your body, inside and out. So it can infect your bones, it can infect your skin and tissue, it can be an intestinal pathogen, it actually can cause infections on the heart valves. So when you think about it, not all of those infection sites are going to be the same in terms of nutrients available to them to eat. The pH maybe, so it might be more acidic or more basic in those environments. There might be other molecules, it might be saltier in certain environments, whatever. And so the question is then, if you consider the idea of nutrient availability, does that mean that cod-Y is really important in all these different infection sites? And the answer to that is no. And what I'm going to tell you is that we have previously worked with an investigator to look at what happens using a mouse model of infection. And what our collaborator out in Ohio had shown was that when you get rid of cod-Y and you increase toxicity on neutrophils, and this is in a laboratory setting, what happens in mice when we use mice as an experimental system to understand how toxic Staph aureus can be. And what we saw was sure enough similar results in that cod-Y mutants, right, can cause more mortality, right, cause more death of the mice, right, in terms of a pneumonia infection, right, compared to the naturally occurring bacteria, which are the ones in black here. So hopefully you can see the difference here in the black versus the hatched bars there. And then further, in skin and soft tissue infections, right, the size of the infection, the lesion, right, is actually higher in cod-Y mutants, right? So that's what this curve here is showing. So this is increasing amounts of tissue death, right? And so cod-Y mutants cause more tissue destruction than the naturally occurring bacteria, okay? So that's all fine and good, right, in skin infections and pneumonia, but it turns out that we did a similar experiment where we put the bacteria in the mouse bloodstream and asked them to infect other sites that the bacteria can infect. And it turns out, and what these data, the only thing I want you to take away from these data is that it turns out that in other sites of the body, in the mouse body that is, cod-Y doesn't really have an impact. And that was really puzzling to us. And so we've been interested in how the bacteria are responding to nutrient availability in these different sites. And one of which is in kidney tissue.
Dr. Brinsmade, sorry, I don't want to interrupt you, but we can't see your screen anymore.
Oh, you've missed my, okay, I'm sorry.
Sorry, I didn't.
All right, no problem. I guess I didn't share it again. All right, and we are running out of time. So what I will tell you is that we are using, hopefully you can see it now, Diana?
Yeah, we can see it now.
Okay. Well, I'm going to tell you, hopefully you could at least imagine what I was saying in those last slides. I apologize. The technology sometimes baffles me. But what I want you to just take home is that we can actually use, remember I told you earlier, we could look under the hood in the bacterium and understand how they work. And one of the things that we do is we use a fluorescent protein that we get from jellyfish that fluoresces when we excite it with certain light. And what we can do is we can engineer the bacterium to turn on the production of that fluorescent protein when under certain conditions. And so what I'm going to show you, right, are data showing that when staph turns on these toxin genes, right, it will fluoresce. And I'm going to fast forward to this image. Hopefully you will be able to see it now. And these are bacteria, individual bacteria in an infection site. And what you can see, I hope you can see, and it may or may not come through on your screens, but this is actually, these are cells from kidney tissue. And if you were to look carefully, you could see lots of little circles, and those are individual bacteria, staph bacteria. And what I hope you can appreciate is that some bacteria are not green, they're kind of dark, and some of them in the middle here are green and are, in this case, they are expressing that fluorescent protein and making that fluorescent protein when that gene, when that toxin gene is turned on. And so I guess there's two things to take home here. One is that we can use genetic tools to reveal what the bacteria are actually encountering, right? What kinds of signals they're encountering in those infection sites. And two, we can learn, so we can do that, and we can learn a little bit about the how the host, your host tissue and host immune system is battling the bacterium. And so in the interest of time, I'm just going to finish up. And let's see. I got to exit out of here. Okay. So what I have told you is that hopefully you're convinced that cod-y acts as kind of this hangry switch in some parts of the body, right? So talked about skin and soft tissue and in lungs for pneumonia, but not in kidney tissue, to turn on the production of these toxins. And the other is that we can use GFP and other so-called reporters to tell us what the bacteria are seeing and experiencing, okay? And that one thing that we didn't get a chance to talk about is that other proteins seem to be working along with cod-y or sometimes overriding cod-y to allow the bacterium to grow and divide and to persist in these different infection sites. And so we do have a lot of work to do, but what I want to tell you is that my job actually is to, in addition to doing science, is to train the next generation of scientists. And so I talked to you earlier about Kevin Malinik and Alyssa, who were Alyssa King, who were two of my first graduate students. They defended successfully their research, and now they're off again in research careers. You can see Alyssa on the right-hand side there. She's giving a talk of her own. And it's a very proud moment when you get to see your trainees actually working and giving talks to a scientific audience. And so I just want to end by saying a couple words of advice, and I'm happy to answer questions if you have any. But work hard and play hard too. So remember that I started out as I still am a nerd, right, and a marching band person, right? So it's important to have hobbies and to do things that make you happy. I'm also a runner and do half marathons. So you can be a scientist and have hobbies and do other things at the same time, right? Balance the two. The other is to identify teachers and mentors whose actions and whose work excite you. Right? I mentioned Dr. Kelly and Dr. Ledbetter, Dr. Escalante and Dr. Sonnenschein, right? They've all essentially helped me get to where I am now in talking with you here. Reach out for opportunities early. So I started my career as a researcher in my junior year in college. Don't wait. Start earlier than that. And I get emails from freshmen and first-year students at universities asking if they can join my lab. And so you should do the same. And the other is ask yourself, what do you want to do, right? What do you want to do? Do you want to have a career in science? And there's lots that you can do with a degree with a science degree and a degree in microbiology. you know, some of them require a master's degree. Sometimes you don't need anything more than a bachelor's degree. It depends on what you want to do. If you want a PhD, right, that opens the doors to teaching and doing research like I do. And the best thing about that is that you don't have to pay for that, right? That if your dream is to do research and earn a PhD, you can do that on with a number of different fellowships and scholarships, and you'll never have to pay a dime for that when you get into a PhD program. And I'm happy to talk more about that. And so I've already talked too long, so I'll pause now. I just want to acknowledge that I have a number of funding sources through the NIH that help fund the research that we do. I didn't get nearly enough time to talk about all the stuff we do, fun stuff. But I also wanted to shout out to those that actually do the science, and those are the students in my lab. They stay the same, and I just get older as time goes on. So with that, I'll pause here. Thanks so much for listening to me, and I'll take any questions that you might have.
Awesome. Thank you so much, Dr. Brinsmade. I think I saw a question earlier in the chat from Kate. If you want to ask your question again in the chat or unmute and ask, feel free to. Let me see if I can find it. And yeah, anyone else who has any questions, please feel free to ask. This here. Let me see. Here, I found it. So, I think this is one that was in context. So is that why if in some cases when you have a bad bacteria in your body, it takes a while for you to discover it?
So, I think I'm not quite, I can't remember exactly the context of that, but what I will say is that I think it's a matter of adaptation. So the bacteria have to, if they get into your, if they're on a surface of, let's say, your desk or your table, or on a, let's say, on the handrails of the bus or the subway car that you're on, they transmit to your skin, and they can get into, let's say, a cut or a breach in that barrier of your skin and cause an infection. But in order for it to cause an infection, it has to adapt to that environment and then multiply and grow. And so, yeah, in a sense it does. Sometimes it takes a while for you to detect and to know that you have an infection, because you don't come down with symptoms. And you only come down with symptoms and after the bacteria have reached a certain number in the body and are starting to produce those toxins as a function of many things, including nutrient availability. I think there was another question that came up in the chat just now. There was a question that asked me if I knew I always wanted to be a scientist, and what really sparked my interest. That's a really great question. And so I think, I mentioned I always was interested in how things worked. I was a NASA nerd. Well, I still am a NASA nerd. So I grew up with the shuttle program. And so when I was in my, I don't know, I was like maybe 8 or 9 or 10, and I would watch the space shuttle launch. And I was always, when I was in elementary and I guess middle school, I always enjoyed going on field trips to the planetarium. And I always was fascinated with science, space, and I don't know, I guess I've always been interested in science. I don't know necessarily if I knew really early on that I wanted to be a scientist. I knew I wanted to be involved in science. And I guess maybe when I was in college and I had that first taste of scientific research, that's when I started playing around in the lab. And I think that's when I really realized I wanted to be a researcher and be a scientist. And that's what spurred me to go on and earn my PhD in grad school. So there was also another question. It looks like you mentioned that if you have a PhD, you don't have to pay for research. What about other degrees? So I don't, I can only comment on what I know, right? And so I think generally speaking in the realm of biology and in chemistry, probably even physics, but I can only say biology for certain. But in biology, and then the different flavors of biology, so microbiology, which is what I'm a part of, but then there's also genetics, uh, ecology and whatnot. Those are other programs or degrees, right, where you often are paid a stipend, a salary to do research, um, and um, and your tuition is waived, and so that way you can, you actually are earning a paycheck. So you're being paid to do science and to get your degree or to earn your degree. So I think that's actually really cool. Generally speaking, that's the way it works for PhDs in the biological sciences and also in chemistry, probably, and then likely in physics. Other degrees in other fields, I think they do it in a similar way, but I can't be sure. And then somebody asked, how did you know you wanted to do microbial pathology? I love bio, but I have no clue what I want to do specifically. And so thanks also for that question. The answer is you don't know until you try something. I kind of, again, I had mentors, so Dr. Kelly, and then also a graduate teaching assistant in Dr. Ledbetter's microbial diversity class. They told me that, you can do, if you liked biochemistry, you could study enzymes in a test tube, or you could actually use microbes to study biochemistry, using them as a model, like as a toy system. And so I guess that's kind of how I decided I wanted to do microbiology research. But then in terms of microbial pathology, it's really just a question as to how do bacteria work and how do they make a living essentially in your tissue, right? And so my lab kind of blends the kind of work that you can do in a test tube with the work that you do in animal models of infection, and kind of understand how these different facets come together to answer questions about how bacteria cause disease. So I don't know if it really answers your question, but I encourage you to be daring and not to do necessarily what's easy or what you are feeling comfortable with and what you already know, but rather have the opportunity to try out new things. When you go to grad school to earn your PhD, often you arrive and then you do what we call rotations, where you test drive different labs, kind of like what I did with Dr. Escalante's lab. And in that case, you can try stuff that you've never done before. And in fact, my department and our PhD program in biology at Georgetown encourages that. And so again, you don't have to lock yourself into a particular area of research. Try out different things and see what you like best, because you're going to be doing it a long time. So you want to make sure you get it right and you know what you like and what you want to do.
I have a really quick question. How long did it take for you to get where you are right now? And how many years did it take to get your PhD?
Thank you for that question. That's great. So my undergrad is 4 years, right? At UConn. And then graduate school depends. So some labs, you can do and it depends on your lab type of research you're doing. For me, it took me about 6 years to earn my PhD, to finish my research as a graduate student. And then another 5 as a postdoctoral researcher at Tufts with Dr. Sonnenschein. So after you finish your bachelor's degree, I guess before you can land your first university researching job like I do at Georgetown, that took about 10 to 11 years. But all the while, right, you're doing fun and exciting research, you're meeting new people, you're learning things. The fun thing is that when you're doing research, you're the first person on the planet to know something, and before anybody else does, right? You discovered that particular behavior or that particular result. And that for me was really cool. And So it actually, even though it was like 10 years, it flew by really quickly. And I can't remember, I can't remember if there was something else, another part